Thank you all for being here. First of all, this is the third and final webinar of this three-part webinar series. For those of you who have not been on the webinars before, or don't remember me, my name is David Hugo, and I'm Sea Grant's South Atlantic Reef Fish Extension and Communication Fellow. As a general reminder, this three-part series is really designed to be an educational resource for anyone interested in the ongoing reef fish research in the South Atlantic. Like the past two webinars, this webinar will start with a guest presentation on a particular research area, then followed by a question and answer session. Another note, if you are interested in any of these webinars or any of the past webinars and couldn't attend or have to pop out for whatever reason, all webinars have been recorded and are being recorded and are available either through the GoToWebinar platform we're using or through Florida Sea Grant or South Carolina Sea Grant's YouTube channels. And then a couple other housekeeping things. So like I mentioned, GoToWebinar is the platform we're using. It automatically muted everyone other than the organizers or presenters. So at the moment, you don't have speaking permissions, but that in the question and answer session, we'll kind of change those to allow people to ask questions. So let's see, what else? I guess before we begin, and I did this last webinar, but this is a Sea Grant webinar series, and we do try to get kind of feedback from you all on the effectiveness of these webinars and whether you all have topics you want to hear more about or whether the webinars met or exceeded expectations. And so I'm just typing in the chat right now just a link to a quick GoToWebinar, or sorry, not GoToWebinar, but a quick Qualtrics forum hosted by the Georgia Sea Grant. So if you all are interested at any point during this webinar, providing a little bit of feedback, it would be much appreciated. Also, obviously, towards the end would probably be better because we haven't really started much yet, but I'll also link a QR code at the very end. That is about it for me. I'll go ahead and introduce our speakers tonight. So we've got two special speakers, Dr. Mark Albins and Dr. Sean Powers with the University of South Alabama. Dr. Albins currently serves as a research assistant professor in South Alabama's Stokes School of Marine and Environmental Sciences. Dr. Powers serves as both director of the School of Marine and Environmental Sciences as well as a senior marine scientist at the Dauphin Island Sea Lab. Dr. Albins and Dr. Powers will be highlighting ongoing offshore research efforts focused on Greater Amberjack, a species that I find really interesting and has given me many sore arms over the years fishing. So Mark and Sean, thanks so much for joining us tonight. I will go ahead and pass it off to you, and I'll kind of descend into the internet world and turn my camera off.
All right, thank you. Appreciate it.
Thank you. So I think I'm gonna go first with a little bit of a background on why we're doing these types of studies, and then Mark will dive into a little more in the details and approaches that we've taken. So I just did want to start with the fact that Mark and I are going to talk about a lot of work, but there's 14 different institutions or agencies working on that, and really a who's who in the world of fisheries management and fisheries ecology science. So we can't go through everybody's specific roles, but I think Mark will talk more and more about the different groups. And even though we're here to talk mainly about the South Atlantic, we're going to talk a lot about the Gulf of Mexico because and South Atlantic because our project covers both of those. Next slide, Mark. So what's the rationale for the study? Well, no one in this audience will be surprised that reef fish management in the Southeast United States has been highly contentious. There's often disagreements regarding stock status and catch levels between the stakeholders on the water and the management agencies. So, and this has come to a point in a lot of areas where it's caused the public to question the scientific basis for many management decisions. And we know that stakeholder buy-in is critical to effective management. If people trust the assessments and trust the science and trust the limits, even if they don't agree with them, if they think they're well informed and to achieve the goals of more sustainable and more opportunity for fishermen, they're more likely to follow those rules. So in response, the US Congress funded 2 large-scale studies to provide independent estimates of what we're calling absolute abundance to help guide future management and build stakeholder confidence. The first of this is one was the Great Red Snapper Count in the Gulf of Mexico. It's the first attempt to go with a independent abundance estimate for a reef fish. And now we're going with the Greater Amberjack Count. I know Will Patterson talked to you about the South Atlantic Red Snapper Count that's funded through a different mechanism. But these three studies really try to get at, do we have the technology now and the advancement now to estimate abundance of reef fish outside of a typical stock assessment? And the reason we want to do that is because most fisheries are managed based on recorded catch. And we know that catch in the recreational fishery is highly uncertain. And even with the best survey designs, it will continue to be highly uncertain. I mean, fishermen tend to not tell the truth about what they catch, whether that's high or low. I'm a fisherman and I rarely tell the truth in what I catch to my friends. And we have faulty memories. It's hard to remember things. There's biases in our memory. And all of these come into the fact that we have often high uncertainty in catch data. Commercial is a little better, but recreational catches are highly uncertain. And most of the species we deal with have a high recreational component. So in fact, but most of the current stock assessment models really rely on the assumption that we know catch. That they really don't work very well if we can't meet that assumption that we don't know catch. And preferably, we want to know the age composition of that catch as well, or the length composition at least of that catch. So that assumption is one that we often have to address. One of the ways to address it is to have an alternative. If you have sufficient scientific surveys that to construct a what we call fisheries independent estimate of abundance, or a relative abundance estimate to help the stock assessment understand what's going on. So I know your first talk in this series focused a lot on survey techniques in the South Atlantic. Those surveys are designed to inform a regular stock assessment that has catch and age data. But even the most data-rich assessments that we do, snapper being a very data-rich assessment, have usually a mix of both. So it still has some of the biases of both. So next slide. So the idea was, can we come up with an absolute abundance estimate to ground truth these stock assessments, or to interpret, or could we manage without a stock assessment ultimately? So what do we mean by an absolute abundance estimate? We mean an estimate of total abundance based on direct observations of fish abundance. So based on science, and Mark will talk about numerous new techniques that have become much, much more cost effective. Dr. Patterson talked about a genetic approach last time in your close kin mark and recapture. So genetics, as well as underwater technology with camera and acoustics have advanced quickly and become much, much more friendly, cost effective. And it's time to see if we can truly get an estimate of abundance. Now we know that it's not a model-free estimate of abundance. It's a different model we're going to use. And one, we think that we'll have less parameters to estimate or less variables that we have to guess at. But we can't obviously count every fish in the ocean. So it's still a model-based estimate, but at its heart is actual count data that anybody could look at our video or ROV data or acoustics and come up with the same count. Ultimately, we have to extrapolate those observations along with the habitat amount that's out there. So why we do that, next slide, is to get an idea of where the models should be, either the catch assessment models or what's the abundance estimate. And again, it's an estimate, but it's based on direct observation throughout the range. So the great red snapper count in the Gulf of Mexico concluded that there were 110 million red snapper in the Gulf of Mexico. And you can see the breakdown by region. And we looked at the number on artificial reefs, the number on natural hard bottom, as well as the number on just sand bottom or uncharacterized bottom. Now this compare this to the stock assessment, the most recent stock assessment, which was about 5-6 years ago, where that stock assessment estimated there were 33 million red snapper. So almost 3 times as many red snapper based on an estimate from direct observations, as opposed to an estimate that looks at the catch data and tries to predict the abundance that can produce that catch. So a big difference in that. And then the good thing is we can also estimate how certain we are about that 110 million estimate. And that's about 11%, a coefficient of variation. So the variability compared to the mean. So we're pretty sure of that estimate we derived. Next slide. So 2.53 times as many age 2+ red snapper in the Gulf than predicted by the NIMF stock assessment. Why? One of the reasons we had to explain is why were the numbers so different? And because we have direct observations, we can start explaining that. When we boiled it down, a lot of the difference in the number of fish we saw with red snapper is these large areas that have very low density of red snapper, too low of a density for fishermen to exploit or exploit very effectively, and areas that weren't covered are just too far away from ports of entry. So the good thing is when you go away from doing catch, you can start spreading out to the whole range. The trick there is how do you use that management advice? Okay, so we think there's 2.5 times as many red snapper. But if you look at what the stock assessment says should be on the artificial reef and the known natural hard bottom, the 2 studies didn't disagree that much. There's about 34 to 36 million red snapper in that. So that then brings up the interesting thing, how do you manage this stock? Because you can't, if you recognize that fishermen are going to only exploit a small amount of that, then shouldn't you manage for that smaller area? Or do you manage for the larger areas knowing that the stock is relatively safe and can rebound quickly? And that's the stage we're at now, is in the Gulf, is reconciling the great red snapper count with the old stock assessment and the new one. Where it's applicable to our question about the South Atlantic right now is, what have we learned from the great red snapper count? Remember, the great red snapper count was the first time we attempted to do this. We thought we could do it. We thought we had the technology, but it was the first time. So we learned a whole lot. We learned, for example, that we need complete engagement with our statistics team. We brought the statistics team in largely at the end of the great red snapper count. For the great amberjack count, they've been embedded with us. They've been constantly talking to us about making sure that the regional estimates are additive, what type of randomization we need to get an unbiased sample. And so in the end, they will know exactly what we've done, so they can come up with a robust estimate. One of the focuses was different areas need to use different gears. Now, this isn't that as big of a problem in the South Atlantic, but in the Gulf of Mexico, it's a gigantic problem. You pretty much have no visibility in the lower bottom in Texas and Louisiana. So you can't use video gear very well. Well, in the eastern Gulf of Mexico, you can use video gear. Acoustic gear has different performances. The different camera systems we're using in the Amberjack Count, they use a different type of camera system, largely in the South Atlantic that you saw, than in the Gulf of Mexico. So we have to really focus a lot more on gear comparisons and understanding what the different gears do in different environments. The other requirement for the Great Red Snapper Count was it was to be independent of National Marine Fisheries Service. So independent of their stock assessment and really independent of their input. Luckily, that's not the case anymore. We were able to involve through a steering committee, National Marine Fisheries Service, so they understand what we're going to produce in the end and have a chance to actually comment. And so that's a really good development. Ultimately, this is the case for the South Atlantic and the Gulf of Mexico. Our estimate will only be as good as the habitat assessment maps that are out there. We, in both the Great Red Snapper Count and in the Amberjack Count, for the most part, we weren't allowed to use any of that funding to map bottom habitat because it would have consumed all of our budget. So a lot of the work we're doing is synthesizing and trying to get the best habitat maps we can. Next slide. Mark?
Yeah, I'm trying for some, there we go.
So I'm going to turn it over to Mark now, and actually he'll tell us what we're doing for the Amberjack, but I wanted to set that mind. That's why Dr. Patterson is doing a study. That's why we did the Great Red Snapper Count. That's why we did the Amberjack Count. Hopefully we're getting better and better at these, but understanding that this is an alternative approach to the traditional catch-based estimates. And what we envision as a team is that these will be two different estimates, and we'll be able to sit down and reconcile which one really best characterizes the fishery and ultimately leads to better management. Mark?
All right, thanks, Sean. Thanks for setting it up. Now we'll get into some of the nitty-gritty details here. So as Sean mentioned, the Greater Amberjack Count specifically builds on the successes and lessons learned from the Great Red Snapper Count. The main goals are, first of all, to provide an independent estimate of Greater Amberjack absolute abundance in the U.S. Gulf of Mexico and South Atlantic using fisheries independent sampling. The secondary set of goals, which are just as important, if you ask me, are to expand the general biological knowledge that we have about the species. We're going to learn so much through this project about spatial ecology, movement, connectivity, growth, mortality, all of these things, important data points, no matter what kind of stock assessment you're trying to do. If you're trying to manage a fishery, these are the kinds of things you need to know. So we're going to learn a lot about Greater Amberjack to inform management decision-making and to address key assumptions of that abundance estimate itself. So for this project, we basically designed it around a multi-phased approach. And I'm going to walk you through those phases real quick just so that you understand kind of how the project is structured. In phase one, we basically, not just our group, but the group that wrote the RFP spent a lot of time and effort gathering stakeholder input. Our group also had as a major goal synthesizing existing fisheries and habitat data. So we wanted to know and have kind of a really good, strong collection of what we know about Amberjack now and trying, to use that to build on. We also wanted to incorporate some new technology. So developing methods like environmental DNA and incorporating those into the different gears that Sean talked about, and making sure that we understand how those gears relate to each other via calibration studies. So all of these things feed into the sample design behind the abundance estimate, right? You have to basically understand what's out there, what you're sampling, understand how your sampling gears work, and then you can define a very efficient and effective sampling design. That sampling design then leads you to your regional surveys, which in this case, like Sean alluded to, we're trying to count fish across a vast range of natural habitats and regions. And each region has its own specific set of pre-existing data sets, its own pre-existing set of environmental characteristics, and we have to work within those constraints. Add to that this idea that if you're trying to count fish across such a large region, you really have to understand how those fish are moving within that region and how those different subregions are connected. So we have a big connectivity and movement component to this study. And that's one of the places where we have leveraged and also taken advantage of as an outreach opportunity, getting stakeholders involved directly in the research, right? So a lot of our connectivity and movement studies, as you'll hear in a moment, have to do with tagging and recaptures. And we rely on the fishing public as well as commercial fishers to help us with that part of the project. All of these different components feed into phase 3, which is actually estimating the absolute abundance of the species. And then that absolute abundance estimate is augmented by basically a full update of the biological parameter estimates for the species. All of these things then feed into, hopefully, the goal is that they feed into our understanding of the species and our ability to manage it, so that they can feed into the NOAA stock assessments and our management decision making. They can also feed into stakeholder engagement and outreach, right? A lot of this project, as Sean said in the beginning, stems from stakeholders not having a lot of faith in the way things have been done so far. So the hope is that we can provide an alternate estimate of abundance that can give stakeholders something to grasp onto and something to have faith in. So the key features of this whole phased approach is, first of all, it's adaptable to differences across regions and habitats. It's scalable from the local scale to the regional scale. And it's efficient in that our sample design is actually based on existing catch data and based on stakeholder input and stakeholder knowledge. So I'm going to go through the main objectives of the study, kind of in order, not in order of importance, but kind of in order of the way we had to do things. So first step is to synthesize habitat data. What does the habitat for this fish look like? What do we know about it, and what does it look like? The main point going into this is that we knew going into this project there are no existing comprehensive maps for the entire region of habitat. So we set out to compile all of the existing sources of habitat data that we could find. And what we found is exactly what you would expect. Some areas are really, really well mapped and other areas not so much. The existing sources of habitat data often have partial coverage, variable resolution, and to compile those existing habitat data sets into a comprehensive GIS product across the entire region was quite the undertaking. I wouldn't say that, we have succeeded, because what we did succeed at doing was finding some serious gaps in coverage in terms of what we know about habitat, especially in the South Atlantic. But we did use, and we were able to use, I think very effectively, those sources that we were able to find. The picture that I'm showing you here is from the West Coast, or the West Shelf, Florida, and basically FWRI has a great data set where they've been doing randomly selected side scan sonar of randomly selected grid cells for a number of years. And so they have not only the ability to tell you what's in those grid cells, but to also estimate, for example, the amount of high relief hard bottom in that entire region. Objective 2 was to synthesize the existing abundance data for the species. So first we synthesize habitat data, and then we synthesize abundance data. So there are existing fishery dependent and fishery independent sources of data for greater amberjack out there. And we basically went out and searched for all of those and tried to put them together into some kind of synoptic product that we could look at. We also know that there's a vast amount of knowledge, not in published scientific papers, not in databases, but in the minds of fishermen, right? So we set out to try and extract some of that information using a pretty extensive survey effort where we went out and collected surveys from fishermen, from the fishing public, on their opinions about where they would go fishing for greater amberjack. And so we've used all of these to inform our expectations in terms of presence, absence, and relative abundance, and importantly, variance of amberjack abundance across the region. All of this leads to the ability to design a very efficient sampling strategy that's region specific and habitat specific. So the idea is that you sample more where abundance and variance are high, and you can sample less where abundance and variance are low. And you can still come up with a very good estimate at the end of the day. And this basically increases your efficiency and makes sure you're not wasting a lot of your precious resources. So objective 3 was to actually estimate the absolute abundance. This is kind of the core goal of the project, and everything else revolves in some way or another around this third objective. And so this objective 3 has 3 main parts. First, the sample design and framework for the absolute abundance surveys, the abundance sampling methods, the tools that we will actually employ to go out and do those surveys, and the calibration of those gears, right? So any project that uses multiple gears to measure something, you have to understand how those gears and what they measure relate to each other. So we devoted a significant amount of effort to that, and I'll talk about that more in a bit. As far as sample design and framework, it's an extremely complex project, and as I said, the sample design is region and habitat specific. So I can't go into all of the details in this webinar, but I will tell you that overall the concept is that we based the sampling design in general on a stratified random or cluster sampling design, where the region is first divided up into these big, large geographic regions. And then within each of those regions, we have 3 primary habitat types: artificial reefs, natural reefs, and uncharacterized bottom. So bottom that doesn't have any 3-dimensional hard structure in it. And the idea is that we wanted to make sure that our sampling gave us an unbiased estimate of amberjack abundance on all 3 of these habitats types across all of these regions. So then we can extrapolate back up to the entire universe. Okay, getting more into the actual abundance sampling methods. What tools are we using in this project? The core approach here for this project is to combine video surveys, and there's multiple cameras that I'll talk about in a second, but they include the 3 main types: stationary cameras, ROV mounted cameras, and towed cameras, to combine those video survey techniques with active acoustics or echo sounder surveys to measure the density of greater amberjack. So cameras have the benefit of being able to do pretty good at identifying species. The problem is that cameras often have an uncertain area of effect. Whereas active acoustics have kind of the opposite problem. You know exactly how much area you're covering, and you can do a really good job of counting fish, but the active acoustics has some challenges when it comes to identifying species. So we'll talk a little bit about how those 2 approaches can come together to give us a robust estimate of amberjack abundance. The specific types of videos used is habitat and region specific, because there are advantages to each of these gear types in different regions. For example, towed cameras are extremely effective for sampling really large swaths of low relief habitat, but not so good at sampling over high relief artificial habitat. ROVs, on the other hand, are extremely effective for sampling higher relief artificial habitats, because you can drive right up to the reef and know what you're sampling. But their ROVs are not so good for covering large swaths of area because they don't move fast enough. And so basically it makes sense to use different tools in different places, but then you have to be able to calibrate them together. We also, as I alluded to a little bit earlier, we wanted to push the envelope a little bit. So a lot of there are a lot of new techniques that are very promising. I think Will Patterson talked to you about close kin genetic analysis as a way of estimating abundance. But we also have some emerging technologies like environmental DNA. Now, this is a technology that's been used a lot in freshwater systems, a lot with species at risk of extinction and species that are in need of conservation, but it hasn't been used very often for fisheries management or for fisheries surveying. So this was actually one of the first, not the first, but one of the first studies where eDNA was used to try to attempt to quantify the abundance of a fishery species in the open ocean. And as you can imagine, there's a lot of challenges involved. So our role in this process is to kind of be a proof of concept for this emerging technology in this kind of frontier. Again, if you're using all these different gears, you have to know how they're related to each other. So a big component of our study is calibrating the gears to each other and to a ground truth abundance estimate. And in our case, I'll talk more about this later, but what we used for that ground truth abundance metric is called a Lincoln-Peterson estimate from a VPS, or a virtual positioning system array. And I'll talk about what that means exactly when I cover our calibrations. Okay, so a little bit more detail on the video cameras. Again, different video camera types for different habitats. This picture shows the three main video tools that we are using in this project. On the left, you see the baited drop camera. These are used for artificial and natural reefs across all the regions. In the middle, you have an ROV mounted camera. In the middle, you have a towed camera. So on the right-hand side with the yellow top, that's USA's ROV, and it has a couple of cameras mounted on it. The ROV is used for counting fish on artificial and natural reefs, specifically in the Gulf of Mexico. And then in the middle, you see the towed camera array. This is called the Seabass system. It's run by Steve Murawski's group out of Florida. And this is the tool that we use to cover those large swaths of uncharacterized bottom across all the regions in the study. Again, dedicated efforts to understand potential biases of each of these camera gears and how they influence the probability of detection. We know that cameras can cause attraction of fish. They can cause avoidance behaviors. The baited cameras, we're adding bait to certain cameras because we want to bring fish into that camera, but we need to answer the question of what would that camera see if there weren't bait in order to use that tool. We are looking at different enumeration methods and how they interact with each other. Different standard protocols that are used by different groups may result in different counts. So we're comparing several of those, and also trying to meet the challenges of the identification difficulties. So as most of you know, a species, the Greater Amberjack, is not the only Seriola in the ocean. It's got some close cousins that are often hard to tell apart. This can be especially true if you don't have the fish in hand. If it's just swimming around and you're seeing it at a middle distance from the camera, it may be difficult to tell species apart. So these are all problems that we've had to deal with, and we've tried to take them on head first and make the best of things and try and learn as much as possible about how to optimize these gears. Calibration studies among these camera gears and coupling them, especially with the active acoustics or echo sounder data, can help to address all of these issues. So I just wanted to show you this. Hopefully the video will play for you. This is footage from University of South Alabama's ROV. And in this footage, if you're good at telling Seriola apart, you may notice that there are 3 different species of Seriola in this image, or in this video, and I'll go back to the beginning here. If you look, the ones with the real high fins are Almaco jacks, so that's an Almaco, that's an Almaco. But then we have some Greater Amberjack mixed in there, and then at the very end you have 3 or 4 right here. This group, 3 or 4 banded rudderfish come around the corner. And so at first glance, these all look really similar, but once you've watched, several hundred videos and talked back and forth with your colleagues, you start to get pretty good at telling them apart. So I just wanted to show you that as an example. Let's see here. Okay, so the great thing about camera gears, like I said, is that you can get good enough to tell the species apart, and most people that can watch a video, it's very repeatable. You can get the same count over and over again from the same video. Active acoustics is very different situation. Basically, if you've ever used a fish finder, if you're a fisherman and you've ever used a fish finder, you basically know what active acoustics is. In our case, we use a much more expensive version than you find on most fishing boats, most small fishing boats anyway. And this can be mounted on the boat, or it can be towed behind the boat with a tow fish like this one. And essentially what you get is something that looks like this, right? You have basically a number of targets along a transect that you kind of cut through the water. So it's like a cross section of the fish that are in the water column. This is all geo-referenced in our case, so we can then make a map of where the highest abundance of fish are across space, as long as we get kind of like mow the lawn, or sometimes do one of these flower or star patterns across a habitat, an area of interest. Then we can make a map of where the fish are. And essentially you can take these data and using a little bit of modeling, you can fit a surface. So in other words, you can interpolate the spaces where you didn't look for fish with something similar to what you did see on both sides of it, and end up with an estimate of the density across that whole space or that whole region. So essentially this is really good at estimating the density, which is an absolute abundance metric of fish. It's not so good at telling species apart. But part of this project and part of the value in the Greater Amberjack Count project, other than the central goals, is that we are pushing the envelope again on these tools. And one of the things that our active acoustics team is doing is trying to get much better at telling which ones of these little dots in this echo sounder are actually Greater Amberjack. The way they're doing that is we're going out and collecting fish, and we're putting them in the CT scanner. And this is a picture of the CT scanner at the University of South Alabama's Children's and Women's Hospital. That's why it's got pictures of fish on it. It's also got fish here on the table ready to go through the scanner itself. Those fish get scanned, and then they create a three-dimensional model of the skeletal structure and all the organs of the fish, especially the swim bladder. And what that allows them to do is to create a mathematical backscatter model that's basically a model of how that fish acts acoustically. And what its acoustic signature or fingerprint looks like. And just because this is really fun stuff, here's a quick animation of an amberjack in the CAT scanner. So you can see that the scanner basically scans through all the different layers of the fish. Those layers are then compiled into a three-dimensional model, which is here in this little video here. And that model models all the different tissues of the fish. The most important ones are the ones that differ substantially from seawater in terms of density. And those are the skeleton, the bones, and of course the swim bladder is the main one. That's pictured in red here in this video. So once they get that, they can actually model the normalized backscatter and target strength of that fish across multiple frequencies of sound. And the idea is that if you use an echo sounder that can shoot multiple frequencies at the same time and get those that data stream back, you can look for the fingerprint of certain species of fish. This is a plot from a paper by Boswell et al. 2020 that shows the species-specific responses for 9 different species in the Gulf of Mexico. And you can tell that if you have a few different frequencies at the same time, you can identify, say, the difference between a gray triggerfish and a red snapper, which is pretty cool. Again, I'm harping on calibration a lot because that's something that we've concentrated a lot on in this project. If you're running all these different gears, you've got to calibrate them together. So we have included in the project some dedicated calibration efforts where we compare our different camera gears. We compare baited cameras to unbaited stationary cameras. We compare stationary cameras to ROVs, stationary cameras to towed cameras, ROV cameras to towed cameras. We also compare active acoustics to all the different camera gears, and all of those gears to a ground truth estimate of abundance, which again, I'll give you a little bit more information about that momentarily. And we compare all of those gears to our emerging technology, environmental DNA. And the goal here is to allow those gears to be mutually informative, to try and learn as much about the gears and how they detect the species from each other as possible. Okay, objective 4, again, like I said, if you get a snapshot of the abundance of fish in a given region, that's all well and good. But if you're looking at trying to estimate their abundance across a wide area, you really need to know how they're moving within that area. So objective 4 revolves around answering questions about amberjack movement, connectivity, and mortality. Currently, the Gulf of Mexico and South Atlantic are managed as separate non-mixing stocks, but we really don't know much about their migratory behavior or population connectivity. So we've instituted a combined strategy, which includes both internal acoustic tags and an extensive acoustic receiver array with more traditional high reward external tags and a big population genetics component. And I'm going to talk a little bit about each of these. But this part of the project is where we really have a lot of opportunity for angler engagement, because we're tagging fish and we're putting them back out there in the ocean and relying on fishermen to catch the fish, see the tags, give us a call and report those tags and their catch details to us. And again, I'm going to talk more about that in a moment, but for this project, to try and encourage that reporting and get that reporting rate up as high as possible, our tags had a high dollar reward of $250 per fish. And Auburn University maintains a hotline as well as an online reporting system. So, you know, I think that hotline is staffed from like 8 a.m. to 8 p.m. 7 days a week. And then you can go on the website and report fish as well. But this is what our tags look like. They're yellow at the base with red at the tip. If you've caught one of these recently or know anybody who has, please give this phone number a call. Okay, so objective 5, again, I've mentioned environmental DNA before, but I want to go into a little bit more detail here. The goal is to investigate the efficacy of and to use eDNA to assess the presence and potentially the relative abundance of amberjack and closely related species. The idea is that ddPCR assays, which that just stands for digital droplet PCR assays, are a technical tool that we can use to basically take a water sample and run it through a machine. And as long as we have the right primers, we can mark different droplets of that water, yes or no. And so a droplet of water gets a yes if an amberjack is present and a no if it's not. And then you can count up the water droplets that have amberjack DNA in them versus the water droplets that don't, and get a relative abundance estimate of amberjack DNA in the water column, which should be at least could potentially be related to the number of amberjack in the water as well. There's a lot of ifs there, and that's something that we're trying to tackle with this project. But the idea is to develop these novel eDNA tools, including the PCR assay, specific to Greater Amberjack, and compare their performance to other gears during our calibrations and the regular surveys in this project. The idea is that we can confirm identification of species, we can estimate sampling versus structural zeros. So in other words, was the fish really there or not, or was the fish not there, or was it there and we just didn't see it? And also to provide relative abundance estimates potentially. We really see this project as a proving ground and a testing ground, and potentially a proving ground for the use of eDNA tools to more broadly study the distribution abundance of marine fishes. Objective 6, again, this is one of those kind of satellite objectives that I think is going to, in the end, have a lot of value added. Updating the biological information for this species. Recent stock assessments have recommended expanded demographic sampling of Greater Amberjack. Age and growth information from the Western Gulf of Mexico especially has been extremely limited. So our plan is to use fishery dependent and fishery independent collections to update the biological information and refine the age length keys for the species. We'll also be archiving samples that can be used with additional funding if it's identified to update the reproductive indices, including fecundity, spawning season, etc. for amberjack. Objective 7, and we've tried to, even though I saved the stakeholder engagement objective for last, we actually have put stakeholder engagement really up front with this project. It's been part of the project from the beginning. We've worked closely with established groups, including the Greater Amberjack Visioning Team, Sea Grant Reef Fish Extension Collaborative, and other groups to facilitate communication and cooperation with stakeholders. As I said before, this started before we were even awarded this RFP. The writing of the research fellowship, or the research program that we applied for, included a whole process where the Greater Amberjack Visioning Team collected stakeholder input and used that to formulate the goals of the RFP. And our funded research project is very responsive to those priorities. So in that way, we're listening to the stakeholders. We've also incorporated local ecological knowledge of stakeholders in our study design to try and make sure that our survey design is as efficient as possible. And all along had active engagement with the for-hire fishing sector to provide platforms for scientific sampling. As I said, the movement part of and connectivity part of our project is highly dependent on commercial and recreational anglers to report those high reward conventional tags. And we also have incorporated a dedicated effort to communicate the results of this research broadly at the conclusion of the study. How do we expect this study to impact stakeholders at the end of the day? It's a large-scale survey. It uses novel integrated sampling approaches. We're leveraging existing data sets. So many of you probably have heard of the SURFs data set here in the South Atlantic. We're trying to leverage that data set along with other data sets that have been collected or are being collected across the region, so that we're not leaving anything on the table. We're trying to integrate those pre-existing surveys into our estimates at the end of the day, to augment our data collection and cost effectiveness. But we're also expecting to be able to provide an independent, robust estimate of absolute abundance of age 1 Greater Amberjack in both the Gulf of Mexico and South Atlantic. We think that this study will result in an improved understanding of the spatial and habitat-related distribution of the species, an improved understanding of their population and movement dynamics in the region, and will help develop an approach and analysis framework. Now, this is something that was started when the Great Red Snapper Count was started, right? We're building on the lessons learned there, and hopefully feeding into the development of a long-term approach and analysis framework that can be applied to future Greater Amberjack abundance estimates and future abundance estimates for other reef fish species. Secondary benefits of the project include estimates of growth, mortality, site fidelity, population connectivity, et cetera, and an improved understanding of reef fish community structure across the study region. Something I forgot to put on this slide, I think one of the other primary benefits of this study is a better understanding of how our sampling gears actually function. I think that our approach is basically going to not just teach us about amberjack, but about how we count amberjack. And I think that's really important. I did want to go through the current status of the project and give you some preliminary results, but with my spiel on top of, or Sean's spiel and my spiel on top of his spiel, we're coming up against, I think, 10 or 15 minutes left in our hour. So this is probably a good place to stop and take any questions if there are any.
Yeah, thanks so much, Mark. Really appreciate that. I think it's really cool how this ties directly into Dr. Patterson's talk a few weeks ago, and there's a lot of parallels between the projects, and obviously this project builds pretty heavily upon what's went on with the Great Red Snapper Count and kind of ongoing with the South Atlantic Red Snapper Count as well. Also super cool, the kind of how cutting edge this is. I can appreciate the CAT scan amberjack. I can't say I've ever seen that before, but it's super cool to see the animation of that too. But yeah, I guess we've got a handful of minutes here for questions and answers, hopefully. Kind of as the last two webinars have gone, it'll be structured the same. Like I mentioned before, everyone is kind of muted right now, but if you do have a question, you have kind of a side panel, and there's a hand icon in that side panel, should be to the right of your screen, and you can actually raise your hand, and my kind of tech staff team and I will go through and unmute you and allow you to ask questions. I'll kind of be calling out people one by one. You are also welcome to type any questions you have in the questions chat as well. I see that there's already been one posted there. We do ask that once you finish asking your question, you just go ahead and remute your microphone. You just press that microphone and it should be red with a slash through it to avoid any kind of distracting background noise. And then last thing, if there's a question that's not answered tonight for whatever reason, please do write it in the chat. I'm happy to compile those and work with Mark and Sean to give you some answers. But that's about it for me. I guess I'll actually kind of kick things off and ask a quick question. So from a stakeholder engagement perspective, I'm really curious about kind of the angler buy-in. I know this project is really focused on whether it be the tagging component or even like the study design. It sounds like anglers were really involved with helping. And what has the buy-in been like? Is there a pretty widespread buy-in from people with the tagging component? Are people excited about that? I mean, $250 is a lot of money, so I would certainly give me an incentive to go amberjack fishing more. But yeah, what's the feedback from that so far?
So far, it's been really good. We've had a couple of meetings that were specifically that. So the process that led to the RFP didn't end when the RFP was published. It actually continued, and after our group was awarded the grant and began working on the grant, after we'd kind of formulated. Well, obviously we'd proposed the research, but after proposing the research and getting awarded the grant, a lot of things changed in our approach just because we had more meetings and talked more and decided, that we could think of a better way to do certain things. But after we had a little bit of time to get closer to what we were going to actually do, we circled back to that group, and there was a big meeting in, this is Kyler Ensign's group, and he invited us to come to a big meeting in New Orleans and basically present what we were planning on doing to a group of fishermen there. And the response was really great. I sat down, I had lunch with these guys, I went out for beers afterwards, and I listened to everything they had to say. And they had some great pointers. I mean, they ended up having an effect on some of the parameters in terms of like, our universe of samples was set at a certain depth, and after talking to some of those guys, we decided we needed to change and look at some deeper sites because we were maybe missing some cryptic part of the population at some deep sites that a couple of fishermen knew about. So we've tried to be responsive to that all along. I think we've gotten a lot of good response in our surveys. We've had, I think 1,800 or more responses in our first round of surveys, and over 2,000 more were planned the last time I checked in with that part of the group. So yeah, at the end of the day, we'll have taken into account the opinions or so of, somewhere on the order of 5,000 fishermen directly, like as data points in our study. So I think that's really cool. And, just anecdotally, my interactions with fishermen have all been really positive. And some of them have expressed skepticism about how valuable that absolute abundance number will be, and I share that skepticism with them. I think that it'll be interesting to see, if we can get the number and get enough, get our variance down small enough that people have faith in that number, and we have faith in it, and it's a robust number. Then what? What do you do with that exactly? And that's, above my pay grade. That gets to the National Marine Fisheries stock assessment guys. So I'm really hoping that there is a way that can be incorporated into the stock assessments and can inform management. But I think that the bulk of the value of this project is actually in a lot of those satellite parts of the project. I think we're going to learn so much from the tagging studies, the genetic studies, et cetera, that we'll basically go from a 1 to a 10 in terms of our knowledge about this species over the course of just a couple of years. So.
Very cool. I guess it looks like we've got a written question. Will you all be sharing those maps that you made? I assume that's referring to maybe the maps Mark had shown in the presentation. I guess Mark, I know I'm not entirely sure what maps we're referring to. Mark Stewart, I guess, do you mind clarifying in the chat what maps you're referring to? I know there were a few shown in the presentation tonight. And I guess in the meantime, while Mark's doing that, like amberjack are obviously a super wide-ranging species, and this is obviously a super wide-ranging project. And Sean mentioned at the very beginning kind of the challenges with a species that has such a wide range. And one of the questions I had was he mentioned that the video surveying isn't particularly as possible in the Western Gulf of Mexico due to the visibility near the bottom being poor, and being a South Atlantic native, I guess, is that just due to the different types of sediment there? I know that in the great red snapper count, one of the things was that snapper were found in kind of this unconsolidated habitat that was actually like muddy habitat. Is that the similar habitat that's causing a reduction in visibility?
It's related, but it's not exactly the same. So here in Alabama, for example, most of the bottom is sand and mud, but we don't usually have what's called a nepheloid layer. So most of the time you can go all the way down to that sand and mud and see fairly good. It's not pretty, right? It's not the Florida Keys or anything, but you can see enough to do fish surveys on most days. But what we have once in a while, and what the Western Gulf is plagued by, is sometimes when the rivers are really flowing, the Great Mississippi and all the other rivers that we're blessed with here in the Northern Gulf of Mexico, when those rivers get going, sometimes they bring a lot of sediment down that kind of gets suspended in what they call a nepheloid layer. There's a different density of seawater there at the bottom, and it kind of holds that really fine clay sediment in suspension. And so a lot of times when you go down to the bottom, you can see fine until you get to within, I would say, somewhere between 2 and 6 meters of the bottom. And then all of a sudden it's just like chocolate milk. And so that presents a little bit of a problem, especially when most of the fish you're trying to count are down there at the bottom on some structure. That's a worse problem for red snapper than it is for amberjack. Amberjack tend to use more of the water column, whereas red snapper tend to be more bottom associated. So I think that we're probably going to not have as much of a problem with that as the red snapper count had. And the idea is that in those situations, our hydroacoustic survey will kind of take over. It'll do the hard work when it comes to those low visibility conditions and give us estimates even in spite of not being able to see very well at the bottom.
Really interesting. Okay, so Mark clarified a little bit about what he meant with those maps. So he was referring, it sounds like, he said, I just meant that you put together quite a bit of location data on good bottom and fish locations when doing the surveying. He's curious if that data will be eventually made available to the public.
Yeah, I'm not sure. That's a good question. Right now, so the RFP that we're responding to with this project did not ask us to produce maps. Obviously, in order to do what the RFP did ask us to do, which is estimate the absolute abundance of amberjack, we had to have a good idea of what kind of habitat was out there. So we had to make some maps and basically collect maps that are made by other people and try and put them together in a way that made sense to us. We haven't gone the extra step that it would take to make those, to put those in a format that would be generally accessible and understandable by the general public. It's kind of a mess, to be honest with you. It's not a mess in that we understand it, but it's a mess in that I wouldn't want to hand it to someone else and expect them to be able to put all the pieces together. So it's a pretty complicated GIS amalgamation of data right now. That wasn't our intention was never to create that for the general public. But that being said, most of the data that we've used to make those maps have come from public sources. And the ones that haven't, we can't really release. Like we've had permission from, certain groups that have done some mapping to use their mapping data for this project, but we don't have permission to then pass those on necessarily. So that would have to be something that is discussed at a higher pay grade than mine, as far as I know.
Perfect. And I guess we've got one hand raised. Kathy Knowlton's got her hand raised. We can go ahead and unmute you, Kathy. And you should be free to speak.
Yeah, hi, good evening. Can you hear me?
I can hear you just fine, Kathy. Thanks.
Sorry, of course my dog is barking. So when I hear you say something like, I think you said 5,000, there were 5,000 fishermen reports and data points in the study, and talking about the importance of the fishermen to this research, I was just curious whether you have published a summary of their impact on this study, like particularly in media the fishermen would routinely see. I think we are always searching for a way to get that message out that's not just in an assessment or in a publication in a journal. And I think this sounds like amazing research that was pivotal on the anglers and the commercial fishermen. So I was just curious about that. Thank you.
Yeah, great question. Thanks for asking it. Absolutely. of course, we're scientists and our job is to publish scientific papers. So that's our main outlet. That's kind of the thing that a lot of us are focused on. But at the same time, we have people in our group who are extension professors, extension agents. Marcus Dryman is our kind of outreach coordinator for the project. And so he is basically developing, he's actually funded through a different group as well to do some similar stuff with the Great Red Snapper Count, but he's directly, he's involved in our project. He's at all the meetings, and he is an extension agent who is creating products for the public as we speak. So all the way down to, our promotional stuff where we tried to get out the message about our high dollar tags. He was in charge of that stuff, but he's also in charge of putting together curriculums and videos and public service announcements and things like that that try to get our message out to the general public and let them know what we're doing with their tax dollars. So yeah, that's a big part of our project. It's also a big part of what Sea Grant does. And so all of our reporting to Sea Grant, they're asking us, how many commercial fishermen have you impacted with this study? How many recreational fishermen have you impacted with this study? And so we're having to, come up with estimates of those and metrics to try and quantify, how many people we're reaching with our messaging. And I think the point that you bring up is a really good one, that we don't just want to quantify how many fishermen we're reaching with our messaging, but how many fishermen have contributed to this work. And I think that is that's a good point, and that's something that I think if we're not doing, we should be, and I'll check.
Great question so far. I guess before, it looks like we've got one more written question. Before I jump into that, Mark, do you mind stopping your screen share real quick? I'm going to go ahead and throw up an evaluation QR code. Perfect. Let's go ahead and see if this works. All right. Oh. Okay, can you all see that? I guess, Mark, can you see that? Okay.
I cannot.
Okay.
It's just the background of your computer screen right now, David.
Oh,
Yep.
Okay. Everyone's got a good picture of me fishing.
Yep. Nice sunset.
Yeah. Let me try this real quick. If this doesn't work, there's no sweat. All right, take two. Any luck?
Yep.
Beautiful.
All right, perfect. Sorry about that. So yeah, as I mentioned at the very beginning, if you all have a minute, just please, we'd love to kind of hear your feedback about these webinars. You can scan the QR code here. I've also linked it in the chat, which is in that side panel. But these really do help Sea Grant kind of evaluate how effective they were and helps refine our ideas moving forward. I know there's a lot of interest in things like this, and we really want to hear from you all about what you're interested in seeing moving forward. So please do take a minute or two just to fill that out and let us know how we did. Without further ado, I'll do one more question here. It looks like Chris Horton has asked a question in the chat. First of all, outstanding presentation. The environmental DNA aspect is really interesting. An index of abundance on any given year would be really helpful with management. How are you accounting for current and tidal drift? Interesting.
All right, thanks for the question, Chris. If I'm understanding you correctly, you're asking how we dealt with water movement in terms of eDNA collection. Is that? I'm going to assume, unless I hear different, I'm going to assume that's what you're asking. And the answer is it's difficult, right? There's all kinds of things. There's all kinds of factors that affect the distribution of DNA in the water column, and it's really hard to measure all of those. And they have to do with waves and currents and all this oceanographic stuff. We didn't really get into a lot of that stuff. We basically measured whether the species of interest was there or not, and we verified that with our camera gears. And then we basically collected water samples from down current of the reef or whatever habitat it was that we're sampling at different distances. And so part of this proof of concept was looking at whether we use Niskin bottles to collect water samples. So you send, this fancy, expensive plastic bottle down a line, and when it gets to the bottom, you can send a messenger down and it closes. And when you bring it back up, it's full of water from that depth. And then you have to take that water and pump it through a filter and take the filters back to the lab and do all kinds of fancy stuff to it. That's kind of an active sampling, and we did quite a bit of that. But what we also found was that passive samplers seem to do a better job of detecting fish. So that active sampling using the Niskin bottles downstream of the reef oftentimes would have 0 when we knew there were fish there, right? So there's no eDNA detectable, but we know there was an amberjack in the water at the time. And so when we started seeing that, we decided we needed to try some different ways of sampling water. And so one of the things, it's kind of crazy, it's a fun story, so I'll take the time to tell you, but one of the things that is in the literature for collecting these eDNA samples is just taking the filter paper or a sponge or something like that and putting it in the water and letting it sit there for a while, and then using that sponge to get your DNA sample. And so we tried some stuff like that. We mounted some sponges and some filter paper rounds to the ROV that we were using for the camera surveys, and as it drove through the water column, it would pick up particulates from the water column, and then we tested those. And it turns out that those passive samplers have a lower miss rate than the Niskin bottles. So the thing that costs a lot that you think is going to be hard to do is actually less effective than just taking a zip tie and tying a sponge onto your ROV and then getting some water out of the sponge. So some really non-intuitive, interesting results have come from that so far. And my guess is that at the end of the day, we probably didn't do enough or spend enough money on that part of the project to actually have pushed it forward enough to use those data in our abundance estimator. But what we have done is spend a lot of time and effort and thought to do some trial and error stuff. And I think that oftentimes those kinds of experiments, even though they may or may not end up being publishable at the end of the day, are going to lead to methods that actually work and methods that make eDNA sampling in the marine environment a reality. And I can just imagine a world in which, we can save a whole lot of money by just going out and collecting water samples if all we really need is presence absence data or something like that. I can even imagine a situation in which we can start figuring out those things like current patterns and how they affect DNA in the water column and get some robust, believable relative abundance estimates from eDNA. It's a big, tough nut to crack, but we got a good start on it.
Awesome, and it sounds like that was right in line with what his question was asking, so you perfectly answered it. All right, let me just check one more time. I don't see any hands raised. No more in the chat. Awesome. Well, that is it for this 3-part series. Thank you all for joining. For those of you who were multi-webinar attendees, I really appreciate it. Thank you to all our speakers from Wally all the way through the end. Mark, I really appreciate you being on. I know Sean has popped off for other commitments, but big shout out to him as well. And like I mentioned at the beginning, these are recorded. These will be up on Florida and South Carolina Sea Grant's YouTube channels. You can also access them through GoToWebinar once the platform has time to kind of process the video. So that might be a day or so, but they should be there. So thank you all for attending. Really do appreciate it. Hopefully you learned something. I certainly did. And thank you again to all our speakers.
Yeah, thanks for the opportunity. Really appreciate it. And thank you all for being here.
All right, everyone have a good night.